Welcome back, nerdlings. In this video, we're going to be discussing tonicity, osmoregulation, and we're going to be going step by step through the water potential equation. Let's go ahead and get started. So first of all, I want to discuss briefly osmosis. So maintaining water homeostasis is so critical for all of living things. Now, osmosis in and of itself is defined as the passive diffusion of water across a semi-permeable membrane. And water always flows from a high water potential down the concentration gradient to a lower water potential. So if we're looking at these U-shaped tubes, which is a very common example that you'll encounter in textbooks as well as exams, you will have a hypotonic solution, meaning less solute, and a hypertonic solution, meaning it has a higher concentration of solute. It could be sodium chloride, it might be glucose or sucrose. So when I say solute, I'm talking about some type of molecule other than water. Now, if we allow that water to diffuse, so we timed it for maybe 10 minutes or 30 minutes or an hour, you'll see that the water flows from a low water potential to a high water potential. So water flows from this hypotonic solution, and the way I always remember it and teach my students is hypo is low and it blows. So it's blowing the water out. Now the other side is hypertonic. Hyper is high and it sucks. So hypo is low and it blows the water out. Hyper is high and it sucks. It sucks the water in. So eventually, after this has sat for several minutes, you will see that the water goes from this lower water potential in the hypotonic solution into the hypertonic solution with a higher water potential. So tonicity is a measurement of the relative concentrations of solute, so again, sugar, salts, between those two solutions, inside and outside of the cell. So hypertonic refers to a solution that has a high concentration of solute and less solvent, meaning less water. So hypertonic, high solute, low solvent or water. Hypotonic has less solute and more solvent or more water. And isotonic means that there is an equal amount of solute as well as solvent concentrations, whether we're talking about intra or extracellularly. So again, hyper is high and it sucks. It has a high concentration of solute. Hypo is low and it blows. Has a low concentration of solute and a higher concentration of solvent or water. Isotonic, we have equal exchange of water across that semi-permeable membrane, and there are equal amounts of both water as well as solute. So tonicity affects cell physiology. The solute moves from the hypertonic to the hypotonic solutions if it is able to. So sometimes we actually can move that solute, but it is dependent upon what type of membrane that it encounters and the properties of the solute itself. The solvent or water moves from a hypotonic or low concentration of solute to a hypertonic solution or a high concentration of solute. So looking at some of the examples here, if we place red blood cells into a hypertonic solution, remember hyper is high and it sucks, the water is going to leave or exit the cell. So the water is going to be sucked out because the external environment is hypertonic. Now, if we place red blood cells into an isotonic solution, meaning that the external fluid has the same amount of solute as the internal environment of the cell, we call that isotonic, and we're going to have a net movement of water across the cell membrane. So the same amount of water is coming in as is going out. Now, if we place those cells in a hypotonic solution, hypo is low and it blows. So water is going to rush into the cell and it is going to blow into the cell. And eventually, because animal cells lack a cell wall, that cell is actually going to lyse. So we do have different types of cells that are adapted for different tonicity relationships. So just because an animal cell is more comfortable in an isotonic solution, it doesn't hold true for plant cells. So let me kind of go through the example below. So looking at this example, for animal cells, it is ideal for those cells to be in an isotonic solution, meaning that there is the same concentration of solute inside and outside of the cell, and a net movement of water that is equal going into as it is out of the cell. Now, whenever we put plant cells in an isotonic solution, they actually become kind of flaccid, so a little loosey-goosey. Now, if we place animal cells in a hypotonic solution, remember hypo is low and it blows, so it will blow those cells up and it will actually cause the lysis of the cell. So if we put an animal cell in a hypotonic solution, that cell will take on so much water that eventually it will undergo lysis. Now, plants actually like this. This is their ideal environment, is to be placed in a hypotonic solution. Now, because of this, it increases their turgor pressure. It makes the cell turgid, which also helps to give cells their rigid, or plants their actual rigid structure and allows them to stand tall without an actual skeleton. And if we put animal cells or plant cells into a hypertonic solution, remember hyper is high and it sucks, both of them will lose water and both of them will undergo damage. So next up, I want to talk a little bit about osmoregulation. So osmoregulation is basically how organisms are going to regulate their water concentration. So I'm going to go through a couple examples on this page. So looking at this single-celled Paramecium, it regulates its water by using a contractile vacuole. So we have contractile vacuoles right here. We'll start off as full, then the pores will open up and the vacuole contracts. When it contracts, it can expel excess water into the environment. So the contraction of the vacuole expels the water. So then we have this very empty vacuole. When the vacuole needs to take water in, it opens the canals. The radial canals start to take up water from the surrounding cytoplasm, and then eventually it will fill that central vacuole and we will go back to having it being very full. So another example of osmoregulation is how fish regulate their blood tonicity. So looking at this, we ask the question, is this a marine fish or a freshwater fish? Now, aside from the fact that you already see that it's designated right here, drink seawater, you could look at the net loss of water. So if it's drinking seawater in, you see that this direction, we have our blue arrows, water movement, and our red arrows are ion movement. So it's losing pretty much the same amount of water and ions right here through the gills. We do have water loss that's occurring through the skin, and so this is pretty much equal. But if you look here, it's losing just a smaller amount of solute than it is water. Now here, whenever it excretes its waste, we have a higher concentration or a very salty urine versus water content in it. So this should be indicative that it is a marine fish. Now another example of osmoregulation are human kidneys. So those little bean-shaped organs, I shouldn't say little, but the bean-shaped organs that kind of sit towards your back, now they help to regulate our blood tonicity. So fun fact, if you think about it, you know, if you're drinking a healthy amount of water, your urine should be a very, very light yellow color or almost clear. Now, sometimes you may not have drank a lot of water, but you might drink a lot of caffeine. So you had 2 cups of coffee. Now, all of a sudden, you know you haven't drank any water aside from the water and the coffee, but all of a sudden you're peeing and it's really, really clear. And you're like, huh, that's strange. Well, it's because caffeine actually inhibits this hormone called antidiuretic hormone. And this is a hormone that basically is responsible for signaling the kidneys to create this environment where it retains enough water and excretes the waste. So without that hormone, our kidneys are just expelling too much of the water. So we can actually get dehydrated if we drink too much caffeine. The same type of situation occurs when we drink alcohol, but if you're watching this video, you should not be old enough to drink yet. So another example of osmoregulation is how animals get rid of their nitrogenous waste. So these are wastes from proteins as well as nucleic acids when they get broken down, and our body needs to get rid of those byproducts. They have to be excreted somehow. And so different animals excrete those compounds in different ways. Now, many invertebrates and aquatic species excrete those nitrogenous wastes in the form of ammonia, which is the most hazardous form. Mammals, such as us, as well as adult amphibians and some marine species, excrete urea. And then we have our birds, insects, land snails, and some reptiles that excrete their waste in the form of uric acid. So an organism's nitrogenous waste form actually reflects how limiting water is in the environment in which it lives. So ammonia is the most toxic, and it has to be immediately diluted and excreted, while urea is slightly less toxic and it doesn't need to be diluted or excreted quite as quickly. Uric acid is the least toxic, and it can actually be excreted as a solid. So moving on to our next component of this lecture, you have to be able to calculate water potential in your AP biology class. Now, a lot of times my students will see the equation and automatically it's like, oh my gosh, there's all these symbols and all these subscripts. What am I going to do? And so I want to take you through a couple of different examples and make sure that all of you are very comfortable with the terminology as well as what each of those constants is representing. So you have to be able to determine how likely it is that water will move into or out of a cell. So we call this water potential, and it's represented by this Greek symbol, psi. So a measure of how likely it is that water is going to move into or out of a solution. Now, pure water, meaning nothing else in it, no calcium, no added flavor, no salt, is assigned a potential of zip zilch 0. So the more negative the potential, the more likely that water is going to move into that area. So the water potential equation is that water potential, again represented by that Greek letter psi, is equal to the pressure potential plus the solute potential. So solute, again, could be any type of salt, any type of sugar. So the units for water potential are pressure units, typically in bars such as torr or milliliters of mercury. If the system is at atmospheric pressure, then the water pressure potential is going to be 0. So water potential of pure water in an open container is 0, meaning that both the pressure potential as well as the solute potential, because it is pure water, is 0. So we would say that the water potential of pure water, meaning there's nothing added to it, in an open container at atmospheric pressure is 0. Now, in biology class, that's probably never going to happen because we're going to be doing a lot of different experimentation, and cells aren't typically in an open container with pure water. They have all kinds of different solutes in them, and we have to be able to contain or to calculate that water potential to determine which direction water is going to flow. So water always flows from an area of higher water potential, which is why this little psi over here is really, really big, to lower water potential, or to an area where there is less water. And so it's going to flow down that concentration gradient. So going to start with small and then kind of work our way up to more complicated equations or questions. So our first question that we're going to examine is, which way will the water flow? Is it going to flow into the cell, or is it going to flow out of the cell? So you should be looking at the numbers. So the internal environment of this cell right here is negative 2.5. So the external environment of the cell is negative 0.5. So this is less negative than this. So because water flows from the higher water potential, or from the lower water potential to the higher water potential, we're going to have the water flowing into that cell. So we had water right here. This is a lower number, so this is a less negative. So the water is going to flow into the more negative space, which in this case is the cell. So water flows into the cell, water will continue to flow in both directions. So water is always going to be exchanged because we do have that semi-permeable membrane. However, that net movement of water is going to flow into the cell itself. So the water will flow from an area of higher water potential, so right here, into the area of lower water potential. This will continue until a dynamic equilibrium is reached. So, an addition of solutes is equal to a more negative solute potential. And in an open container or in an animal cell, the pressure potential will be 0. In turgid plant cells, the cell wall can actually exert pressure onto those plasma membranes. So now we're going to look at the solute potential. So this one gets a little bit more complicated because we have several different factors that we have to take into consideration to actually calculate this. Now, whenever you are working one of these problems, you will be given what you need. So it's pretty much a plug and chug type equation that you have to plug your numbers into to get the correct answer. So for our solute potential, we have solute potential equaling negative ICRT. So I stands for the ionization constant of the solute. So for example, the ionization of sucrose is 1. The ionization of sodium chloride is 2, so on and so forth. The C stands for the molar concentration of the solute itself. This is a constant, or this is a factor that will actually be given to you. And then R stands for the pressure constant. This will remain the same, and it will be 0.0831. And then temperature is always going to be represented in temperature Kelvin. And so you're always going to be given the temperature. So it might say that the temperature is 10 degrees Celsius. And so if you were plugging and chugging into this equation, you would add the 10 degrees Celsius plus 273 to get that temperature into Kelvin. Now, let me kind of go through each one of those little parts. So I, again, stands for that ionization constant. So for sucrose, I equals 1 because it does not ionize in water. However, for sodium chloride, the ionization constant is 2 because sodium chloride ionizes when it dissolves and becomes 1 positive sodium ion and 1 negative chlorine ion in an aqueous solution. Now I know you're already like starting to have that little bit of like anxiety build, like, oh my gosh, this is what we did in chemistry. So if we're talking about ionization constants, typically in any type of word problem, you will be given that information. So in that word problem, it would give you something along the lines of sodium chloride has an ionization constant of 2. So you won't technically have to do that specific step, but I like to make sure that you understand how that number came into play. So we're going to practice that skill now. So the question is, for magnesium chloride, what would the ionization constant be? Now again, take a breath, because you are not going to have to do this particular step in class. However, again, I just want to make sure you understand how that ionization constant was calculated. So if you see that I equals something, you understand how you got there. So magnesium would ionize into 3 different ions if it was dissolved in water. 1 magnesium ion and then 2 chloride ions. So C stands for the molar concentration of the solution itself. So the molarity is the moles of solute over the volume of the solution. And again, typically you're going to be given the molar concentration. You're not going to have to calculate that out yourself. R is the pressure constant, and again, this is always going to be a given, and it is on your formula chart from the AP Biology equations chart that you can have on every single quiz and every single test that you take in this class. So T is represented the temperature Kelvin. So Kelvin is the temperature in degrees Celsius plus 273. So again, if I gave you that little information tidbit of we're at 10 degrees Celsius, a little bit cold, and I want to know the degrees in Kelvin, you would add 10 plus 273, which would give you 283 degrees Kelvin. So quick question, room temperature is about 20 degrees Celsius, so what would it be in Kelvin? So you would just add 20 to 273, and you would get 293 degrees Kelvin. All right, now we're going to start kind of putting all of these different components together. And again, remember that you will always get that formula chart on your AP Biology test, so you do not need to memorize this equation. A lot of times you're also going to be given your ionization constant, your molarity, and then that pressure constant is always going to remain the same. All of those will be given for you. So you are given this scenario. If a cell's pressure potential is 3 bars and the solute is negative 4.5 bars, what is the resulting water potential? So again, you'll have your equation. Water potential is equal to the pressure potential plus the solute potential. So water potential is equal to our pressure potential, which was our 3 bars, and our solute potential, which was negative 4.5 bars. So our water potential for this scenario would be 1.5 bars. Now moving into a slightly more complicated problem. So a cell with a water potential of a negative 1.5 bars is placed into a beaker with a solution with a water potential of negative 4 bars. Now, will that water flow into or out of the cell? So the answer is that the water is going to flow out of the cell from the area of higher water potential, which will go down the concentration gradient, to an area of lower water potential, or negative 4 bars. Now, the way you might want to think about this is water is going to flow to that higher solute, all right? So it's flowing down the concentration gradient of water, meaning in that hypotonic solution, we have less solute. In the hypertonic solution, we have a higher amount of solute. So the water is going to flow into the hypertonic sucks. So water is going to flow from hypo is low and it blows. So the water is flowing into that hypertonic solution. Or water is flowing out of the cell from an area of that higher water potential to the lower water potential. So next question, what is the water potential of a 1 molar solution of sucrose and an open container at 20 degrees Celsius? And again, you would always be given the equation. So the answer is going to be negative 2.4 bars. So to set that up, you're going to have your solute potential, and you know that that equals negative I CRT. I is the ionization constant. And so we know that I is equal to 1, or the ionization of sucrose and water. Sucrose does not ionize, so we say that it has an ionization of 1. We are given the molar solution, so we know that it is a 0.1 molar solution. So we plug that in here, and this is our C. R is a pressure constant that we are always given. So again, we've basically just plugged that in. Now to get the temperature in degrees Kelvin, we are told that the container is at 20 degrees Celsius. So we added 20 degrees to 273 Kelvin, and we got 293 degrees. Now we multiplied all of that out, and we got negative 2.4 bars for that solute potential. So we plug it in here. So water potential equals 0 plus negative 2.4 bars, which is negative 2.4 bars. All right, so our last question. This is something more along the lines that you might see on your AP Biology exam. You might be given a scenario like this where it says, determine which of the following solutions will gain the most water if placed into a sample of pure water in a piece of dialysis tubing at the temperature indicated. So we know that pure water has a water potential of 0. There's not going to be any solutes in that either, because it gives us that it is pure water. So we're given different solutions. We have sucrose, which we know has an ionization constant of 1. And then we also know that the tonicity is given, all right? We have 2 molar. So we have our concentrations, and it even gives us our temperature, so we don't have to calculate out temperature. So in this instance, we would basically just plug and chug. And so for each of those, for the first one, we have our ionization constant of 1. We have a molar concentration of 2, which again, if we go back, is given right here, and it also gives us our temperature. So we are always given our pressure constant, which will always be 0.0831, and that will be given in your formula chart. And again, the temperature right here, 298 Kelvin, is given right here in the chart. So again, it was not anything you have to calculate. This is straight up a plug and chug. So we multiply that out, we get negative 49.53 bars. And we will do the same for each of the other two substances. Now, looking at the numbers themselves, this one is the most negative. So if we go back to the question, it says determine which of the following solutions will gain the most water. So which one of those solutions is water going to flow into? Water is going to flow from a higher water potential down that concentration gradient into the lower water potential. So meaning that it would have a higher concentration of solutes. A, after we calculated it, would have a higher concentration of solutes. So the answer would be A. So I hope that that was helpful for everybody. For more videos like this, you can hop on to www.nerdlingscience.com. This is the Queen Nerdling signing off for today. Have a wonderful day.